This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 226, recorded on March 12th, 2013. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. The introduction to this episode is a bit different. A few weeks ago, I had scheduled to record a TWiV with Terry Dermody, and when he arrived in my office, a.k.a. the TWiV studio, Dickson Despommier was there, and Terry looked at him and said, "Oh my gosh, one of my favorite medical school professors." So they sat down and began to reminisce, worked as quickly as I could to set up the recorder, and captured them in the middle of a sentence, I believe. So what you're going to hear now is a conversation already in progress.
Have a single foundation year that's sort of combines normal and abnormal human biology, right, and then a single clinical year, and then let the students choose a major. And in the major, the students will have the opportunity to pick elective courses that represent, in most cases, synthesis of both basic science and clinical medicine. And I think a course like yours might not work at Vanderbilt because we don't have enough international patients that would come back. But my gosh, in New York, so every single internist, and see, they would know that they were going to do internal medicine at that time. Right. Every pediatrician, the pathologists, some surgeons, especially these surgeons who have, the Nigeria lust, and so, and they go off, right? I mean, they go off and they work and come back. Incredible things, and all of a sudden they, what the hell was that? They pull out, and so then you have a course not of 160 Columbia medical students, but maybe a course of 40 or 50. But these dudes are in it like no other dude you've ever had. And then in the clinic, they're seeing travelers, international clinic. We have a really good.
It's not about you; it's about me.
Put your thing on.
Yeah. Anyway, okay. But we have a good travelers clinic and a good refugee clinic. But I don't know if we're busy enough to do it. I'm just trying to think. Maybe we could hire you.
Who? Dickson? Hey, don't break my virus, Dickson.
I've been.
You already broke it once. You broke a receptor.
How could you break that little virus?
He knocked it off.
It was easy.
Mm-hmm.
Are you okay there?
Plastic.
Are you okay?
Yeah. This is the virus they're trying to make extinct?
Yes.
Mm-hmm.
See, he broke a receptor off.
Oh, my God. Mm-hmm.
There was a wonderful physician here while I was a student here from Vanderbilt. He happened to be African American and one of the few that has ever been at Vanderbilt. I think up until recent past.
Yeah.
And he was an advocate of Harold W. Brownism. Go abroad for 2 months or 5 months and see what it's like, and then come back and your eyes are like saucers.
Yeah. After your course, one of my most formative experiences at Columbia was during my senior year. I grew up in Ithaca, New York, and of course I went to Cornell, and then I came here for medical school. And my worldview was shaped entirely by upstate New York and by the bubble of being a medical student in New York City.
Right.
And I really thought I needed to learn something more about the world. And I'm not good with language, and so I needed to go someplace where I could speak the language at least well. And so I went to Ireland, and I spent 2 months in Ireland. The March and April of my fourth year, I worked for a month in a hematology unit. In another month, I was a precept with a general surgeon. And, that experience for me was transformative. You come back and you care for lots of people who are really different, even though they might speak the same language. Not that they thought they were speaking the language that I did, but
that's right.
You appreciate these things that unite us as human beings in a different way. Yeah. And I show those slides. I took a bunch of pictures when I was there, and I show those slides still in my virology course now as an advertisement. You all need to get out of the bubble of medical school and your worldview shaped by suburban wherever, often near a coast, with lots of privilege growing up. None of it's their fault, just to expand their worldview and see a little bit more of what makes us all human. Anyway,
You
okay.
grew up in Ithaca?
I did.
Is that right?
I did.
I went to Cornell also.
I know
You
that.
do know that.
Yes.
So you went to Cornell.
Mm-hmm.
So you went to high school in Ithaca. Mm-hmm. You went to Cornell for college.
I did.
Then you came here to PNS for medical school.
I did.
Wow. And then we overlapped maybe 1 year.
Mm-hmm. Mm-hmm.
Right. Which time you were in Steve Goff's lab, correct?
Yeah. When did you come here?
I came here in September '82.
Oh, we overlapped for 3 years, but
Okay.
you were an assistant professor getting a lab started.
Yeah,
and,
I didn't see you.
And I was a resident and trying to figure out, how to manage.
So you're already
these.
a resident at
Yes.
that point. So you
Still?
you matched here at Columbia.
I did.
In
And
which service?
What?
Medicine.
Medicine.
Yeah.
Okay.
So I was both a student and a resident here.
Wow.
So graduated in '82 and then finished my residency in '85.
And this was a research residency? Not really?
No, the physician scientist training then was quite different than it is now. So that now research-oriented medical schools have large MD, PhD programs that might enroll 10 or 20 or even 30% of their student body. These would be students who are committed to a career in discovery. They understand that early on and earn a PhD degree. And then we have all kinds of pathways for physician scientists in residency and fellowship to develop skills and more patient-oriented research as well as basic research, including PhD degrees. Lots of master's programs, a lot of institutions have it. But when I was training, medical school was very traditional. There weren't a lot of opportunities for research. We had one free summer, and that was between the first and second year of medical school. And I was trying to put myself through school as best I could with help from Columbia and help from the federal government through loans. So that summer I took a break to work, and I was a room service waiter at the Regency Hotel for a whole summer. That was good fun.
Very interesting. That's medical training.
Well, it's a different kind of training.
Oops, I'm sorry.
Good morning, room service.
Wow. I didn't know that.
So, and then our residency, this is the pre-Libby Zion days. And so, I think back to those days. As interns, we weren't allowed a day off until after Labor Day. We started the 1st of July. We didn't have a day off until after Labor Day. And the idea was that we couldn't take a day off because the services were so busy that an intern couldn't cover your service if you were off. And so we did have opportunity for research, and some took it, but that didn't come until the senior year. And we had about 5 or 6 months of elective time that a lot of my colleagues spent a month on the various consult services. And I, at that time, had already decided to go to Boston. I was going to work with Bernie Fields. These were formidable virologists. I was not at all a virologist, and I thought I needed to gain a little bit of experience before I went to work with Bernie. So, gift from God, Steve Goff decided to take me into his lab to work for a few months, and that really worked out well.
Did you know that he worked with Steve?
I just found that out.
So it was for a couple of months, that's it?
Yeah, no. Well, I started, I think, in the January of my senior year, and I worked until May or so.
This is when you were still in medical school?
No, when I was a resident.
Okay.
I'm sorry, I said senior year,
Yeah.
but I meant senior year of residency, so
Okay,
my third year of residency.
And they worked you pretty hard, didn't they?
Well, during that time, you were worked very hard as an intern. You were on every third night, except when you were on every other night, and that was pretty much the intern schedule. Juniors were on every other for about a third of the year, and then every fourth for the remaining 2/3. And seniors were largely on every sixth night. That's what we did. And during those elective times, you were on 1 and 6, supervising other residents. So you were up, but it wasn't that strenuous. So I really had a very good experience in Steve's lab.
So when you came to medical school, was it with the intention of doing research at some point, or you didn't know at that point?
Yeah, what a good question. I think I came to medical school largely because I wanted to be a teacher.
Really?
Yeah. After my, those first thoughts of a career fireman, astronaut, and so forth had faded.
Baseball player.
Yeah. Well, I had no talent, no athletic talent at all.
That hasn't stopped a lot of them.
I can keep track of my numbers, but that's about it. Throwing the ball over the plate is a challenge for me. And so my parents are both teachers. So my father taught in the hotel school, actually, at Cornell.
Oh, that's a great school.
And my mother was an elementary school teacher. She taught 4th grade. And so those are, my earliest role models. And we all stand on the shoulders of those giants who are our parents. And so I thought it would be fulfilling to teach in the context of a medical school. I thought that would be fulfilling work. I never envisioned practicing like a practice where you would see patients all day and go to the hospital when they were ill and care for them. I thought I would be in a school. I just enjoy that environment. At Columbia, the infectious disease physicians, by far, were just terrific educators. Harold New, Glenda Garvey, Mark Goldberg. These were wonderful, wonderful people. And I really enjoyed that they cared for patients of all ages, both genders. They weren't restricted by an organ system, and they knew lots of medicine.
That's right.
And they knew lots about biology. And I thought, wow, what a way of applied biology, infectious diseases, and just watching evolution happen in front of you. You didn't have to wait epochs to see new things happen. It was there in one patient. You saw it.
And the very beginning of the AIDS epidemic.
Oh, well, talking about, I'll get back to this in a moment, but talking about life-shifting events. So the very first cases of AIDS were reported in December of 1981. That was the December of my 4th year. And, of course, I stayed here at Columbia to train in New York, Los Angeles, San Francisco, Boston, Philadelphia, big coastal cities, the epicenter of the AIDS epidemic. And we saw so many young people, at that time almost all of them men, who presented with very late-stage HIV, with profound immune deficiency and life-threatening and life-taking opportunistic infections, and that really defined a lot of our careers. And by the way, I mean, talking about humbling, many of us pursued a career in infectious diseases because we thought it was like medical surgery. So what we would do is we would elicit the history of the bathing in the Nile, which might explain why the urine was red, and that would suggest a diagnosis of schistosoma hematobium. And we would make the diagnosis with a urine for ova and parasites, and then prescribe the praziquantel that would treat that and render the patient almost back to perfect sense of a whole. Complete normalcy.
That's right.
And then along came HIV. And despite our best efforts, the vast majority of people we cared for didn't survive. Even though we might be able to ameliorate to some extent their condition, we certainly couldn't cure it. And sadly still can't, although we have retroviral therapy that, of course, is much more effective, keeping people alive for long periods of time. But that was a career-shaping epidemic. Sure. And then, all of us who went into this field thinking we were going to cure everyone realized that we're going to have another and really important objective in our careers, which is to help people sometimes with that last chapter of life, something we didn't plan on, but that's what we were called to do. Well, anyway, that interest in infectious diseases led me to think about, of course, training in infectious disease. And
so do you think that AIDS kicked that in part for you?
Well,
or not really.
I had decided I was going to do infectious diseases before HIV.
Okay.
But I can tell you that I think the hubris of I'm going to be able to cure everybody I meet, that AIDS took that away, and I think made all of us who approach the field with that feeling, I think, much more effective physicians because of it. So my interest in infectious diseases took me to a number of programs and to meet a number of people who are engaged in infectious disease research. And I must say, I was captivated by Bernie Fields and captivated by the reoviruses. And I thought, well, I'm going to work with this man, and I'm going to work in this lab, and I'm going to try and figure out how the reovirus is home.
So you learned about Bernie by reading. Right.
Sure. Again, it's amazing when you tell young people these stories. I mean, imagine life.
You mean me? Like me?
Like you, Vincent. Imagine life without the World Wide Web.
It's hard to believe.
Or podcasts. Or Facebook or Twitter. I mean, just imagine. And so how did you learn about people? Well, you read journals that they published in. Right. And it was very important to publish in journals that people would read, because if you published in a journal people wouldn't read, even though it might have been a good journal for another field, people wouldn't see your work.
That's true.
And the way you found out that something was published, you remember these days, you went to the library,
of
you
course.
pulled out the Index Medicus.
Yeah, yes,
And
of course.
you thumbed through it and you found, what you were looking for, and you saw the papers published in that area, and then you'd go and get a cart and you'd go to the stacks and you'd pull out the volumes and you'd Xerox the papers you were interested in reading.
Absolutely.
That was a lot of fun, actually. I loved the library.
I loved it. I don't know. I shouldn't tell this. This is my Bernie Fields story.
Sure.
Did I ever tell you? So when I met Bernie, I was in Peter Palazzi's lab. I was a second or third year PhD student, and Bernie had come to give a seminar. So he walked in the lab one day, and he went to talk to Peter, and I think he said hello to, or Peter introduced me to him. And then on the way out, he said to Peter, "Yeah, I think we have it licked. I'll let you know what happens." And then he left, and I said to Peter, "What is he talking about?" He said, "Oh, he thinks he can figure out why 12 segments are packaged into the Riovirus virion in the next couple of months," which we still don't know.
I think that's fair. Right.
But I was impressed as a brand new student. Wow, he's going to figure this out, because even I had been working on flu, so I knew this issue with flu. How do you get the segments in? So that's my first. And then I used to read his papers. Absolutely.
Yes.
People who worked on segmented viruses read each other's papers. So you learned about Bernie, and then you decided after you finished your residency, you were going to go work. Was that a postdoc, basically, or something?
With Bernie.
With Bernie.
Yeah. So infectious disease fellowships then and now are largely focused on, of course, training in the clinical subspecialty of infectious diseases, but also training in research. When I was training, the idea was that the majority of infectious disease specialists would practice in academic settings. The majority of them would support themselves through a scholarship, through research, that infectious disease was a hospital-based career where you would care for very ill patients with infections. There really wasn't a career option for an infectious disease physician in a community setting, right. Either in an outpatient or inpatient kind of
Is that still the case?
Care. Well, that was another change with HIV, because now we have a lot of people who are infected that are in care, and most of the care is provided by infectious disease physicians who are practicing in the community. So, in fact, adult infectious diseases, the field or discipline that one would pursue after internal medicine training, is either a 2 or a 3-year subspecialty program. The 2-year program would be for those who know they want a practice-oriented career, whether it's outpatient or inpatient practice, and they might do antimicrobial stewardship, infection control, hospital epidemiology, but they're largely going to do practice, and there are lots of opportunities for those people. Or a 3-year subspecialty, which incorporates 2 years of a research focus, which in my opinion is just long enough to let someone know that they have maybe a little bit of talent and a little bit of enjoyment for research, but nowhere near long enough to be sufficiently well trained to be competitive as an independent investigator. One needs a lot longer than just 2 years. So now we see probably in adult infectious disease, the majority of trainees are going to pursue careers that will be primarily clinically oriented. And in pediatric infectious diseases, it's probably still a bit more academically oriented than clinically oriented. But that just, I think, reflects the fact that there are not that many opportunities for the clinical practice of pediatric ID short of an academic setting.
Right.
One really needs to be based in a university in order to practice pediatric ID.
And now, of course, that's what you do. You're actually chief of pediatric ID.
Right.
And maybe we'll find out how you got to that point. But now here you are in Bernie Fields' lab. You're doing an infectious diseases fellowship, which means you take care of patients, but you also do some lab work?
Right. So the way that the fellowship worked out was that the first year was predominantly work in the hospital. And so I split my time largely between the Brigham and Women's Hospital and the Beth Israel Hospital, and spent a couple of months at Boston Children's. And that was a pretty intense year. There weren't opportunities for research. I may have visited Bernie a couple of times working on grant applications, postdoctoral fellowship applications, but no time to really have a project of any kind. And then that first year ended, and then the new first year started up, and then we no longer had clinical jobs. Then we were in our labs beginning the research component of our training. So, imagine Bernie's lab at that time had about 20 people. They were superb. wicked smart, wicked creative.
Who was there?
Wicked hard.
Was Ken Tyler?
Wicked
Skip
hardworking.
Virgin?
Ken, Tyler, Skip Virgin, Barbara Sherry, Leslie Schiff, Linda Morrison, a couple of the students, Lori Sturzenbecker, David Wessner, Kevin Coombs was there. It was just a formidable group of people. And I'm the brand new person, an MD without much training, and I can remember just making buffers and not knowing how to operate the pH machine. Or, when it was my responsibility to split the lab cells, we kept mouse L cells we used to propagate reovirus in these big spinner cultures. I mean, they're amazing cells. They'll grow if you spin them. They'll grow if you plate them. And we kept a couple liters of these that the lab would use for plaque assays and purifications and so forth. And we all took turns splitting them. And there was a lot of anxiety in the lab when it was my turn to split the cells. And the anxiety was largely initiated when I found the research assistant who maintained the cells daily and said, okay, now, I've drawn this grid of what a hemocytometer looks like. Now, if I find the cells in this square, what does that mean in terms of the concentration? And then how do I get it down to 5 times 10 to the 5th per mil? And everyone thought, oh my God. We've hired somebody who was promoted from about the 3rd grade into medical school and now taking care of cells in our lab. So, but I have to tell you, if it wasn't for Rhonda Basil-Dubey, who was a postdoc at the time, and Max Neibert, who was just finishing his PhD and was going to stay on as a fellow with Bernie, they took me under their wing, and I must say they taught me an awful lot of science, and they also taught me how to write and how to present, how to think through data, and I owe a large debt of gratitude to the both of them because Bernie was busy. He was the chair of the microdepartment at Harvard. At that time, he was already a member of the National Academy and had a lab of 20-some people. And so to spend a lot of time helping a brand new MD fellow learn how to do dideoxy sequencing and how the ladder actually came to be the ladder. How did the G's know that they were in that lane? How did that work? That was not something that was on his agenda in any particular day or week. But with Rhonda and Max, they really helped an awful lot.
Cool. What did you work on? Do you remember?
Yeah. Oh, of course. So I'm the scientific lineal descendant of Rafi Ahmed.
Wow.
And so Rafi had made observations when he was a graduate student with Bernie about how viruses and cells co-evolve in a carrier culture to form a persistent infection. And essentially the way it works is that reovirus is cytolytic. It's going to kill cells. And so if cells are infected at either a very low MOI or a high MOI passage stock is used to initiate the infection, after a week or so, most of the cells in the flask are going to die off. But if one aspirates about half or 2/3 of the medium and resupplies with a little bit of fresh medium, these little islands of cells would start to propagate, almost like a little archipelago, that would be like, Hawaii. And you would see them come up and eventually populate the flask. And after they got about 25 or 30% of confluency, then you could trypsinize them and passage them and make a line. Now, if you quantified the infectious virus in the supernatants of those cultures, it was substantially high, 10 to the 7th, 10 to the 8th PFU of reovirus per ml. And if one wasn't careful in taking care of those cultures and let them get overgrown, you could propagate them for years. And so what Rafi found is that essentially the cytolytic reoviruses select for resistant cells, and the resistant cells in turn select for mutant viruses that are capable of infecting the resistant cells, although not as fit as a wild type virus would be for a wild type cell. So this keeps it in balance. These studies of Rafi were published in 2 Cell manuscripts and then a number of other Journal of Virology and Virology papers that he had as a graduate student. Incredibly productive. Really hasn't slowed down, Dr. Ahmed, since he was a graduate student with Dr. Fields. Yes. In any event, so my project was to figure out what the nature of the mutations in the cells and viruses that led to this coevolution that occurred.
Sure.
That took us about 15 years to do, but the project actually started started in Bernie's lab, and one thing we knew at the time is that mutations were selected in one of the viral gene segments, a gene segment called S1. S1 encodes the attachment protein of reovirus, which is this long fiber. And the idea was make a lot of mutants and sequence their S1 genes, and then you'll figure out, because we know something about this protein it encodes, what the nature of the mechanism of the adaptation would be, because the mutations will point you to a functional domain, and then the eureka moment will come when it's clear that that's the domain that's altered. So this was 1986 and 1987. Mm-hmm. So in 1985, the first S1 gene sequence was published by Rhonda Basildoubi. It was a letter in Nature, like a report in Nature. One S1 gene, 1.5 kb, and I mean, amazing. And so sequencing double-stranded RNA, that was thought to be rocket scientist. I mean, who knew it was rocket science? But that I was going to do it. And so to get me ready to sequence all these mutants I was going to be making from the persistent infections, I was, suggested, Max and Rhonda thought it would be a good idea to just sequence some S1 genes from field isolate strains of reovirus, strains collected from all over the world years before I got to the lab. And one of the great serendipities, I have to say, is that in working up those sequences, we found several strains that were incapable of binding to sialylated glycans, to sialic acid, which most reovirus strains use as a co-receptor. So these were sialic acid blind viruses that were selected in nature. I don't know, and I just happened to sequence them. But the advent of those SA blind, sialic acid blind reoviruses allowed us to do an amazing amount of science afterwards, because we needed viruses that didn't bind to sialic acid in order to identify definitive proteinaceous receptors for the virus. And if we never had these sialic acid blind viruses, we never would have found a proteinaceous receptor for reovirus. It's just, the serendipities that come in science are amazing to me. What great luck.
It's huge.
Absolutely.
It's important. Yeah. And it's important to do things that you don't know why it's important to do them, but they may turn out to be important someday.
So Louis Pasteur was right.
Yes,
he was right.
Yes.
So you learned how to do science in Bernie's lab? Is that fair to say? Yeah.
I think it's fair to say that I learned that I really enjoyed science. Yeah. That I really enjoyed it. I started to appreciate important questions. I started to. I learned that writing was really important and learned some basic skill in writing. But I have to say there were big gaps in my professional development when I left my postdoc.
How long was that?
Well, I was there for 4 years, a little over 4 years. And, you asked me how I came to pediatrics. And, one day, I just have to tell you this story. So, all of these stories,
I'm looking at your publications with Bernie Fields here.
So, Margot and I had our sons in Boston. They're now 23 and 25, and this is the spring of 1990. And so the boys are 2.5 and 6 months, something like that. And we lived in Jamaica Plain in a condo in a beautiful Queen Anne Victorian. We lived up on the 3rd floor. The laundry's in the basement. One car. It was hard. And now Margot's home, raising the children, and she's a hotel salesperson, and now she's home raising the kids. And those 5 o'clock calls came, and when are you going to be home? And I'm moonlighting every other weekend because Margot had always had a greater salary than I did, but now she's home, and so now I have to moonlight to pay the mortgage and moonlight to pay off my medical school loans, and Boston's just expensive. And so one day, Bernie walks into the lab room where I worked and puts a recruitment letter on my desk. And it's a letter from a man called Peter Wright, who was my predecessor as division director at Vanderbilt in pediatric infectious diseases. Peter, he's now at Dartmouth, wonderful person. He's worked on almost every virus.
I think polio too, right?
Exactly. Strongly interested in polio and rota and flu. And anyway, so the letter was a recruitment for a basic scientist, a PhD scientist, with interest in microbial pathogenesis to come to Vanderbilt and to establish a new research center that had been been established by the Lamb family. George Lamb was at the time a retired CEO of the United Parcel Service, and so they had made a very large gift to the university to establish this basic research unit. And the idea was that this unit would be a component of the Pediatric ID division, Infectious Disease division at Vanderbilt, which had, even at that time, almost a 20-year tradition of excellence in clinically oriented research around vaccine evaluation. But what was sought was some complement that was much more fundamental. And initially the idea was the Lamb Center will develop the vaccines, and then the clinical investigators in pediatric ID will actually test them. That was the initial concept. So Bernie puts the letter on my desk, and he said, "Terry, I know you're not a pediatrician, but this is sort of interesting. And you might just give it a look."
What were you, though? You were an ID person?
Yeah,
by then?
I'm an internist, an ID-trained internist.
So you had done a year of ID at Harvard, right?
And then before that, 3 years of internal medicine residency at Columbia.
Okay.
And so now I moonlit a ton and saw a lot of children in my moonlighting. And I worked for the Harvard Community Health Plan, and we had these terrific nurse practitioners, pediatric nurse practitioners that taught me a lot of pediatric care. But I was by no means a pediatrician, and just little short exposures to pediatric infectious diseases. So anyway, I took the letter home and I showed it to Margot, and I said, I'm pretty flattered. Bernie thinks that maybe I would be competitive for this job. And Margot said, would you pick up the phone and call that Peter Wright and let him know that you will be down there tomorrow to interview if he would have me? So off I was, and it was an interesting time in that. The late 80s, early 90s were a cyclical decline in NIH funding. Pay lines were maybe not quite as low as they are right now, but they were low. And there was a lot of concern about the future of science and the future for physician scientists. And all I will say is that the training that I received in Bernie's lab and my interest in trying to answer basic questions, fundamental questions about microbial pathogenesis, the question that still keeps us up at night is how viruses home. Once they infect at a primary site, they navigate in many cases to a site of secondary replication, causing disease at that site. And how does that work? How does the homing work? That's what keeps us up at night. And the idea that that fundamental interest might lead to lots of discoveries around the general area of infectious diseases and cell biology, immunology, biochemistry was attractive, and I had some options. But for me, the Vanderbilt opportunity was just unsurpassed. And so there I was in 1990, joining a pediatric infectious disease division.
Was that the only job you looked at?
I looked at 3 others, and but that was the one that was clearly the best. And not to say that the others were not good; they were. But this for us was the right city, it was the right setting. Virology at Vanderbilt at that time, Vince, was really modest. The department of micro and immuno had just recruited a new chair, Yatsik Hauger from Harvard. There was a lot of recruitment that was going to go on. The medical micro course needed some work. The dean at the time would say it needed a lot of work. I didn't have a large number of virology colleagues, but knew that they were going to come and knew that I could play some part in recruiting them. I felt in teaching, I was only limited by my ideas. We could fashion whole new courses, and it was, well, let's just say we had so much opportunity to improve that anything we would have done would have been better. And so...
you could be the teacher that you always wanted to
exactly,
be. Right?
And all of those options were available at Vanderbilt and wouldn't have been available to the same extent at some other really wonderful institutions that I looked at.
So you went to the pediatric ID division immediately?
I did.
Is that right?
I did.
As an assistant professor?
I did.
Okay.
Yeah. I'll come back to one story about AIDS, and because this really was another career-shaping time for me, in that I enjoy caring for patients. In fact, still do. I do about anywhere between 4 and 6 weeks a year on the clinical service in pediatric infectious diseases. I don't do any general anything anymore, and I don't do any outpatient work. I strictly focus on inpatient consults. And so when I joined the pediatric ID division, I thought, Well, these are my new partners, and I'm going to help on their clinical service if they would have me. So if they would teach me, train me really, how to do pediatric infectious diseases, then I'll split my clinical time and I'll do a month or so in pediatric infectious disease and a month or so in adult ID. Now, this is the early 90s. Now, it's 5 years before highly active retroviral therapy was used. And we understood, we at that time had a handful of antiretroviral drugs, but we knew that they all gave about 6 months extension of life, but none of them were going to maintain a long survival. And we had two services on the adult ID side, a traditional ID inpatient consult service, and then essentially an HIV service where all of the patients we admitted were HIV infected and all of them had significant opportunistic infections. And I that's the service I worked on. I worked on the HIV service. And it was very hard work and very sad work. But I thought it was really some of the most important clinical work I've ever done. I just felt it was so important to help the last chapter of a story be told. And in many cases, these guys, most of them were men, would come home to Nashville largely at the very end of their life. They left the city where they had their career and would come home with an incurable illness, essentially to die. Many of them were gay, and sometimes, especially for fathers, this whole concept that their son, 25, 30, 40 years younger than them, was coming home to die with a disease that was contracted because he was gay and not accepting any of these things. And I spent so much time with families, largely with fathers, just sitting and listening and talking, pulling a lot from my experiences in Ireland, what unites us as people, helping with that last chapter, just that last little bit. And that was hard work, but I thought somebody really needed to be in there. And there are many of us who felt in the very same way who did that. And then in the mid-'90s, with new drugs, we actually closed that ward.
Wow.
Because, we were treating these guys, and all of a sudden their loads are suppressed, their CD4 counts are moving upward, the opportunistic infections, not that they're melting away, but we're not seeing them, and we can treat things now. So the ward closed. And what I realized is that what I enjoyed most about that experience was being a family physician, taking care of families. And that's what pediatricians do. They, of course, take care of children, but they take care of families. Yeah. So it was about 96 or 7 when one of my colleagues, Kathy Edwards, took a sabbatical and was going to be off our clinical service. And I thought, well, I could donate another month on the adult ID service, but that would mean that one of my partners would have to work doubly or triply hard. So I'm going to do all my time in pediatric ID. And that was maybe 96 or 97, and that was the last time I saw adult patients in a formal kind of way. So now I only do pediatric ID when I work clinically.
And what's the most common thing you see in pediatric ID in Nashville?
Well, I would say that bone and joint infections caused by methicillin-resistant Staph aureus, pneumonias of a variety of kinds.
Ears.
Okay, so do we see otitis media? We do, but not usually in infectious diseases, only because pediatricians are superb infectious disease physicians. So we're only called to see sick children in the ICU with meningitis, encephalitis, pneumonia with empyemas. We see a fair number of nosocomial infections, not many, but we do, like catheter-related bloodstream infections. We also see a lot of infections in immunocompromised people. It is amazing what we're doing now with transplant. The stem cell transplants, the solid organ transplants, and of course, to maintain those transplant allografts, in many cases, one needs to suppress a T-cell immune response. And so, a whole pharmacopoeia of infections now can present, and the differential diagnoses are quite long for the different possibilities.
Right.
Yeah.
So when you went to Nashville, besides being in pediatrics, you wanted to have a lab, and you started that at the same time, and for that you had an appointment in a basic science department. Right. So was that important for you to do?
Well, here we've been spending all of this time talking about clinical medicine, and I must say I've never spent more than about 15% of my professional effort focused on clinical medicine. I always thought that that at least for me, I would be most fulfilled leading a research team and teaching and training. Those would be members of my team. And so to be a member of a basic science department was absolutely essential for me. So I joined the Department of Microbiology and Immunology at Vanderbilt, and thank Jacek Kauger as the new chair for inviting me to be a member of his faculty. And I would say that Jacek gave me every opportunity to teach and train that I could have asked for. And still, I lead the virology section of the medical microbiology course, and with Chris Aiken, I direct the molecular virology course, which is a graduate level virology course. So those are my two teaching responsibilities now. I've been doing both of them for now over 20 years.
So Dickson, if you reach behind you, see that little black box that's right on the edge next to the virus is right there. Take that. I thought I had the medal I got when I visited you.
Yes.
You were not there very long,
No.
but you started a visiting professorship called the LAM
Lecture.
and I was the first LAM
Exactly.
professor, and I came and visited you and gave a talk, and it was a lot of fun. That's the first time I had met you, is that right?
Yes.
Because even though we overlapped here, I never saw you.
Right.
Did you see me walking around ever? No, probably not.
If I did, I couldn't have picked you out of the other scientists in Steve's lab. If you thought my eyes were big when I joined Bernie's lab, I mean, imagine when I was in Steve's lab. That was a completely different time, and I was a resident, knowing that I was just visiting Steve's lab for a few months, and I would still wear the white polyester pants that we did as residents. I often wore a bow tie then. I wore clogs. It was just a different kind of an era, the mid-1980s. There we were.
So when you went to Vanderbilt to start your lab, you brought, I presume, this persistent Riovirus project, because you said you worked on it for 15 years.
Right.
Was that your main focus, or were there other things?
Yeah,
too?
we started, continued both of those lines of inquiry, both the trying to sort out the mechanism of the persistent infections and working on really structure-activity relationships of the attachment protein, trying to understand domains that bound to receptors, what the receptors were, functions of the receptors and pathogenesis. And those two themes, the latter one continues. It took us a very long time to understand the nature of the alterations in persistent infection, and that turned out to be exciting, because we knew for some time, really through the work of Lori Sturzenbecker and others who preceded her in Bernie's lab, that Riovirus requires endocytic proteases in order to uncoat. The uncoating was known to be acid-dependent, and so the assumption was that acid-dependent proteases or acid activation of the particle to render it a more suitable substrate in some way was required for the uncoating. But the identity of these proteases was not clear. So what we found is that the mutant cells selected in the context of persistent infection had alterations in the activity of two endosomal cathepsins B and cathepsins L. They're both cysteine-containing proteases in the papain family, and so the hypothesis was, because of the alterations in the cells, that maybe those cathepsin proteases were in fact the enzymes that encode the virus. And so Dan Ebert, who was a graduate student in my group, showed in fact that is the case. And he showed this by using genetically ablated cell lines and specific inhibitors of these cathepsins, and also showed that purified cathepsin could act as an enzyme to generate the disassembly intermediate in vitro. That was all that was necessary in order to do that. At the time that Dan did his experiments and identified these cathepsin proteases, B and L, as critical for uncoating the virus, there were no viruses known to require cathepsin proteases for either entry or for processing of glycoproteins. And in following on the heels of Dan's work, it was rapidly shown that Ebola glycoprotein is processed by cathepsins in order to uncoat. SARS spike protein is processed by cathepsins. Becky Dutch showed that the Hendra and Nipah glycoproteins have to be processed by cathepsins too, but they're processed on the way out, and so they get processed as the particle is assembled. And so the work that Dan did, identifying these cathepsins, I think in a significant way established a foundation for these other studies that showed a function for cathepsin proteases in entry.
So are those lysosomal based?
They are.
Because I spent 4 years at Rockefeller as a postdoc, or 3 years, I should say. But one of them was in Christian de Duve's lab, so he was isolating the lysosomes and finding out what their role was in physiology and medicine. Of course, and 2 years later, he got the Nobel Prize.
Yeah, of course. I mean, it's an amazing story because imagine the virus has selected a pathway that the cell normally uses to process antigen for presentation in MHC class 2. Right. So it gets. I always think about the endocytic pathway as the subway, and the virus wants to get out at the right stop. It does not want to fall asleep and wake up at a different stop. That might not be good. It just might not be good. So the virus has chosen a route that antigen is normally processed, and as soon as the processing is complete, before all the way down to the lysosome, the virus can penetrate the endosomal membrane and escape. I mean, it's amazing. Now, the story is also very interesting with regard to these host proteases in a very new application for reovirus, which is the use of the virus as an oncolytic. In other words, a natural mediator of cell killing of cancer cells. And so it turns out that these cathepsin proteases in many cases are elaborated into the extra tumor microenvironment where proteolytic activity is quite rich. Mm-hmm. So when reovirus is delivered to a tumor, it actually uncoats extracellularly, and this is a much more efficient way of initiating infection, and this is probably one of maybe even a number of ways that a tumor is more susceptible to a virus than a non-transformed tissue.
So when it's digested by the protease, it still has the receptor attachment protein, or it gets in some other route?
Yeah. So there are two steps in the disassembly. In the first step, which is a proteolytically mediated step, the attachment protein remains.
Okay.
The main outer surface protein is removed, and the protein that's left behind, that's the inner surface protein, changes its conformation and is capable of penetrating across a membrane. Now, when that disassembly occurs, then the fiber is shed, the outer capsid is shed, and what is liberated into the cytoplasm is just the core particle.
Right.
And what's so remarkable about reovirus is within the core, of course, the genome segments are there, but the polymerase is there too. And the polymerase is never free in the cytoplasm. It's always in this core. And it goes to work to catalyze the transcription of those gene segments, and the RNAs, like, little angel hair pastas, come out of the core vertices, and then they're translated, of course, in the cytoplasm.
You were going to say something.
Well, no, I mean, just thinking about how lysosomes actually work, though, because they throw in a whole bunch of acid hydrolases, right? Including DNases and RNases and everything else. So how does this virus escape from that particular part of it?
Well, it's a good question, and I would say that the virus doesn't escape in all circumstances.
Okay.
And I think the virus escapes in few circumstances, which is one of the reasons why the particle-to-PFU ratio of reovirus is as high as it is. Entry is not efficient. And so it may take anywhere between 10 and 100 infectious particles to get one of them actually to enter and infect a cell. And the rest of them, we think, either attach and are released free, or attach and uncoat, but don't make the membrane penetration step very efficiently and move right down to the lysosome where they're degraded. We think, in fact, that at least in some contexts, that aberrant uncoating can release the genome, which is never thought to be released. I mean, double-strand RNA is a potent activator of all kinds of innate immunity. But we find that, there's nothing that activates the RIG-I, IPS1, IRF3 pathway like reovirus double-strand RNA. It is really, really good.
So that's a mean of evasion, keeping the RNA in the particle, right?
Absolutely.
And what extrudes is single-stranded.
Right.
Those are translated and then encapsulated before they get double-stranded again, right?
Right.
So it's brilliant.
It is. It is. It is.
You couldn't make this stuff up. That's what I always say.
Yeah.
Now, the cathepsins are lysosomal. Mm-hmm. So the lysosome is fusing with the endosomes as they come in, right?
Yeah.
So Rio really goes farther into this
absolutely.
subway. It goes further on the A train than any other virus almost.
All the way to Rockaway.
Yeah. We did just one story on this that illustrates sort of the depth of the endocytic pathway that reoviruses penetrate. This is a deep stab into the center of the cell, and we've learned this through a number of strategies. One, to identify the organelles within the endocytic pathway that are required for functional encoding, and we know that the virus has to track to organelles marked by Rab7 and Rab9 that mark late endosomes and lysosomes. This is work of Bernardo Mino, a postdoc in my group now. Bernardo also did recently a drug screen where we screened the NIH clinical collection. This is a group of drugs that have all been used in humans, so the toxicity profiles are all known. And we did this to see if we could learn new things about reovirus replication. And what we found is a dozen or so drugs that were active, but a half a dozen of them all inhibit microtubule function, like docetaxel and Vindicine and so forth. These are mostly chemotherapeutics, and they're quite active in this regard. And so we wondered, well, firstly, are they just intoxicating the cell, and that's why the cell's not making virus? And they don't. We did a really clever experiment because you can generate these disassembly intermediates in vitro. We call them ISVP, for infectious subvariant particle. And so we infect the cells in the presence and absence of drug with virions, whole virions, and these virions have to traffic through the endocytic pathway and uncoat deep in the in the late endosomes. The ISVPs attach and they penetrate either right at the cell surface or just beneath the cell surface. So we find that the microtubule inhibitors inhibit infection if we infect with virions, but they don't inhibit infection if we infect with the ISVP disassembly intermediates. So something that differs between the two is where the drugs are working. So Bernardo is a brilliant microscopist, and he chases particles right down microtubule tracts and can see them and silence the motor, the dynesin motors that move the particles into the interior, and of course that blocks infection. And then we did a control experiment with another virus that we've been working on recently called Chikungunya.
If you're Italian, you'll know all about that one.
Well, Chikungunya is a great virus.
My wife thought that was a dish. I think I'll have some chikungunya.
Every once in a while, people come up and they say, Chikungunya. Where do you keep the chickens?
Or the gunyas for that matter.
Yeah, there you are. Well, Chikungunya has its name from the Mikunda dialect that's spoken largely in Tanzania. It means literally bent over or bent up, and it describes the arthritis that this virus causes. It's an alpha virus. It's vectored to humans by mosquitoes, and it causes these explosive outbreaks of arthritis, largely in East Africa, in the islands of the Indian Ocean, in India, Southeast Asia. But you're right. I mean, every once in a while, an infected person will get back to Europe. And in the levels of virus during chikungunya virus infection in a human being are so high that if a mosquito, and these are Aedes aegypti or Aedes albopictus, these are common mosquito species.
He's speaking my language.
Well, they are. And if they bite you and you're viremic with chikungunya, that mosquito is going to be fit then to transmit that virus to your neighbor.
So were the reservoirs?
Oh, absolutely.
No animal reservoir.
No, animals can be infected, but humans certainly can be reservoir hosts for chikungunya. It's not like West Nile, which is out in the birds, and we're just incidental hosts. Yeah.
You should sign it.
Oh, I will.
He wrote a book on West Nile.
Well, there you are. Yeah. Well, anyway, back to entry. Chikungunya, like alpha viruses, requires low pH in order to encode as well, but it doesn't have to go down the subway tract as far.
It gets out before the proteases, I guess.
It does.
Right?
It gets out in early endosomes. So where the microtubule inhibitors will inhibit reovirus because it has to go from early to late, the microtubule inhibitors do not inhibit chikungunya, which of course is another indication that the drugs aren't just toxic.
I bet they don't inhibit polio either.
Probably not.
Because that gets out pretty early in the endosome, depending on the cell, of course. So do you think this, now reovirus, I remember you saying once it does cause some human infections, but not a lot.
Right.
But the cousin, which you do not work on, rotaviruses, those are the human pathogens. Is all of this that you have learned about uncoding also applied to rotaviruses, cathepsins as well?
Yeah, it's an interesting question. It doesn't appear that rotaviruses require proteases in order to encode. And there's a lot of controversy in the rotavirus field now about the entry pathways that are used. And, this is in general. There's a lot of controversy in the field about the entry pathways used by any virus. And I think it reflects a couple of things. Depending on the cell type, the virus may have options. And if one uses a very well cell-adapted strain of virus, that virus, if you close down one route, is going to find the alternative pathway and going to gain access into the cell interior. So you might falsely conclude that I've inhibited clathrin-dependent uptake and the virus gets in, so the virus doesn't require clathrin. No, the virus would like to use clathrin, except that you've left options open, and those options are reasonable ones. It's clear that the protease inhibitors that we use to inhibit reovirus infection, that would also work for Ebola, for example, or SARS coronavirus, they don't work to inhibit rotavirus infection. And most think that entry occurs early, like polio or chikungunya. May or may not require a low pH step. That's unclear, but it certainly doesn't require proteases in order to encode.
Okay. So, part of the idea of the uncoding by proteases is that in the gut, this could participate in virus entry.
Right.
So this could work for Rio, but not for Rota, you're saying, most likely.
Well, now, in fact, fascinatingly, both reovirus and rotavirus require proteolytic processing in the intestinal lumen in order to infect.
Okay.
And so for reovirus, that ISVP form of the virus is actually generated in the gut lumen. And if you block that, this is work that was done by Bernie Fields and postdocs, Dinah Botkin, who work with Bernie, and Dorsey Bass, who also collaborated with Bernie in this work. And they found that if you treat mice with protease inhibitors and infect with virus, well, they're not going to get infected. But if you generate the disassembly intermediate in vitro, they'll infect a protease inhibitor-treated animal. Right. Now, ditto with Rota, but the processing is different. Rotavirus, has these spikes called VP4. And there are many copies of VP4 on the particle, and those VP4s have to be processed by protease, and the cleavage products remain particle associated, but it's almost like flu HA. It has to be proteolytically processed in order for infection to occur, and so and that occurs in the gut lumen.
Okay.
Yeah.
I want you to tell us two stories. I have a couple of things that we have to cover here. I got it all in my head, Dickson.
Good.
Now, besides having both gone to Cornell,
yes,
you and I have had parallel careers in a few other ways. No, good ways.
This is a good old boys meeting here?
Our listeners are going to learn some science.
Oh, okay.
Dixon, you know that the reason I got this job because I had made an infectious DNA clone of polio, right?
Right.
And then when I got here, I cloned the polio receptor.
Right.
This man has identified the receptor for reovirus, or a receptor, and he has made the first infectious clones of reovirus.
Oh, wow.
Correct?
Fantastic.
This is correct.
Now, you don't have to make a transgenic mouse because you already have mice susceptible.
So Rio's a mouse-specific virus, just so I can straighten this out.
Actually...
what does it infect?
Yeah, mammalian orthoreoviruses, the viruses that we study, appear pretty well adapted to all mammals. And in fact, the prototype viruses that we use in the lab were isolated from human hosts, from children actually. This virus that's been trademarked Riolysin is the one in the same type 3 Deering that is used by Riovirus labs.
Now, Patrick Lee has started that company.
Right.
And we actually talked with his CEO, Brad Thompson,
remember that
on TWiV a while ago. Remember that?
A while back.
So, did Patrick train with Bernie Fields?
Patrick trained with Bill Yaklik.
Oh, okay, the competitor, right?
Well, in a way.
They were buddies, though, right?
It's a small enough field that it's like Cheers Bar. I mean, everybody knows everybody's name. And so, but there was a time, I'll never forget this, it was the ASV of either, it's 1989 or 1990.
It's okay. We forgive you.
Yeah, 1989, 1999. Roy Duncan, who's a friend and colleague, was a postdoc with Patrick, and I was finishing my postdoc with Bernie. It could have been 1989, and we had back-to-back oral presentations at ASV where we both presented the same abstract.
Really?
It was the S1 gene sequences of prototype strains of the 3 reovirus serotypes and an analysis of those sequences to learn something about how the protein might fold up in space. And it was
results?
pretty. Yes, we did. Yes, we did. Published them about the same time. And so, but that T3D, the D is Deering. Deering was a child in Cincinnati, and that virus was isolated by Albert Sabin in 1955. That's where type 3 Deering came from. And so
he named them Rios, right? Respiratory enteric orphan?
Orphans. He felt bad for them. it didn't cause a disease. And so what we know is that epidemiologically, most mammals appear to be infected with reoviruses. Most humans are infected before they go to the first grade. And we know that from 3 or 4 epidemiologic studies now published, including one from Nashville. When these oncolysis trials are done, virtually all the patients treated with reovirus therapy have reovirus antibodies preceding. So, I mean, everybody has been exposed to the virus. Disease associations are modest. The virus has an exquisite age restriction for disease. It will only produce disease in the immediate newborn period. So for a human being, you have to have the bad luck of being born to a mother who's never been infected with reovirus, so can't passively protect you. You have to be infected by, someone's, person who's shedding reovirus at the time that person is holding the child. And the reovirus probably has to be a strain that has a given tropism for either the nervous system or bile duct epithelium or the heart, where reovirus-reported disease has been suggested to occur. So this would be, triple bad luck. And so I think that's why disease association is so unusual.
So is most exposure very, very young, or does it continue throughout?
Oh, it continues through life. But what we know is that most people will have developed an antibody response before they go to first grade.
Terry, maybe it's beneficial.
Well, okay, it's interesting to think that, and you wonder. You talk about an egalitarian virus. I mean, one that infects all mammalian species. Yeah. Its gene segments appear to be equally fit for all of the species that it infects. It doesn't appear, there are some, in laboratory settings, preferred constellations of genes that like to stay together during reassortment type experiments, but when you generate phylogenetic trees, the segments are completely non-joined. There's no relationship between those phylogenetic trees. And disease association is limited. It doesn't really occur with most animals in which reoviruses have been linked to cause infection. So this is, it's a perfectly egalitarian virus, fit to infect everybody, but leaves everyone happy.
Yeah, well, I bet someday we'll find that it's beneficial. You know Skip's idea that viruses can help you from his herpes virus infections of mice, and I bet a lot of these we have. And Rio is not the only one that infects many people. A lot of these single-stranded DNA viruses infect most of us. There are a lot of polyoma viruses in us as well. Yeah. And of course, we're all full of herpes viruses. You look skeptical, Dickson, but you have probably 2 or 3 herpes viruses right now.
Probably 5.
5. Oh, I have to revise what I tell everyone.
Most humans have 5.
I've had warts.
Simplex 1.
VZV,
EBV, CMV,
HHV6.
6 in your telomeres, right?
There you go.
What's your eyes bugging for, Dickson?
I was thinking back to something that Terry said before about oncolytic activity, and the presence of antibodies already there. Does this not present a problem for that strategy?
Right. Well, thus far it has not. And there was a paper in Science Translational Medicine last year that was focused on human subjects treated with reovirus oncolytic therapy. And the main thrust of the paper is that much of the virus actually adheres to cells or infects cells to disseminate hematogenously. And so that it's not exposed to the neutralizing response that's there. And then it homes so exquisitely to these tumor cells and infects them that it's not circulating the bloodstream for very long.
Any success rates to talk about?
Absolutely. There's much more information, of course, by the firm, and where all of this is posted on their website. But reoviruses now are in phase 3 trials. It's the most advanced trial that I'm aware of for the treatment of refractory head and neck cancers. And so it gets to phase 3 because they're encouraging results in phase 1 and phase 2. And as far as I know, I'm not involved in this research at all, but as far as I know, the initial results are encouraging, so much so that they're continuing to enroll patients because they want to get larger numbers and so forth. Yeah,
Thompson told us at the time that it was looking good, and then we have several listeners who were really interested. And
it was a while back.
They periodically send us updates, and it does look very good. And as you said, they're in phase 3.
Yeah. Now, one point, just last one about reoviruses and pathogenic potential. The mammalian orthoreoviruses, the ones that we study, they're not fusogenic. And so what they do is they infect, enter, uncoat, and complete a replication cycle and leave a cell by either lysis or some egress mechanism that we don't understand. That's actually a focus of new work we're doing in our lab. How does the virus get out? Not to say that we fully understand how the virus gets in, but we thought we'd have a look at how the virus gets out. But there are fusogenic orthoreoviruses, and the prototypes would be the avian reoviruses. These viruses all encode a little protein that's called a FAST protein, which stands for fusion active small transmembrane. This small open reading frame was discovered by Roy Duncan and his colleagues, and Roy has been working on the function of these fast proteins for some time. Now, they don't just infect birds, these fusogenic orthoreoviruses. They infect lots of species, reptiles and insects and humans. And there have been reports in the last couple of years. I would ask, but it's too easy a question. What mammalian species do you think has led to the transmission of a new virus into humans that causes pulmonary disease?
What species?
Take it, things.
Wait. What species has led to transmission of a virus?
Of a new virus to humans that causes
pulmonary disease?
Pulmonary disease.
Lung disease.
Lung disease. A pneumonia.
Birds.
Well, they have wings, but they're
Chickens.
They're mammals.
They're mammals.
Bats.
Of course.
Bats, of course.
Of
Yes.
course.
20% of the mammals, they have to be there.
Yes. So it turns out that
Yeah.
bats, in addition to being infected with every other virus that we know of and a whole bunch of viruses that we don't know, I'm quite sure, are also infected with fusogenic orthoreoviruses, and they're now starting to spill into the human population, and there have been several case reports of humans with pneumonias, including human-to-human transmission. Just a few cases, one family, but it just gives rise to the idea that maybe reovirus does have pathogenic potential, and we ought to probably be focusing on how these viruses replicate, so that if it does, we'd have some,
It's a good try, Terry. Good
arrows in our quiver.
try. Very good try.
You read the
the
significance of
corona.
this grant application, it's all
I
about.
think the coronas are getting all the attention these days.
Well, there you are. And then there's new SARS-like viruses coming back in the Middle East.
What about the ones killing the pigs in China? I'd like to know more about those.
Well, those are the ones where they threw the pigs into the
Yes.
river upstream of Shanghai. Did you hear
2000.
about this?
Have you heard
Thousands
much
and
about
thousands
this?
of pigs dying from what they think are circovirus, porcine circoviruses. But
And the farmers are getting rid of them because they're just chucking them in the river and they're shooting right down in the middle
and
of
apparently Shanghai gets its drinking water from the river, so people are very upset.
Apparently they do.
I'd heard that the bats. So there have been a couple of metagenomic studies in bats, and I'd heard that they found Rios, but I didn't know there was some spillover
Absolutely.
into people. We'll have to check that out. I just want you briefly to describe for our listeners, because even though these are both published stories, they're beautiful. Let's say first, how you clone the receptor, identify the receptor, a receptor for reovirus, because more than one, right?
Right.
There's
right.
sialic acids, right?
Right.
And then there's a proteinaceous one so tell us the strategy, because people will like to hear it. It's cool.
Well, thanks for that question. So it's clear that reoviruses bind to more than a single receptor. The vast majority of strains bind to glycan. We've now identified glycans bound by both type 1 and type 3. They terminate in sialic acid, but they're different. Different gangliosides that terminate in sialic acid serve as the glycan moieties. We think that glycan binding is sufficient to place the virus on the cell surface. It's a low affinity interaction, but it's very avid because of the abundance of sialic acid, and then the virus can surf until it binds to a definitive high affinity docking receptor that then can initiate the process of infection. So, it was about 10 years that I was at Vanderbilt before I thought we were in a position to take a big risk and try and identify a receptor for reovirus. Many receptors for reovirus had been published by very good people and just hadn't stood the test of time as bona fide docking molecules that led to virus infection. And so, in fact, the Bermuda Triangle of Virology is littered with wonderful receptor candidates, many of them published in coffee table journals that just didn't make it, And so it really took a bit of courage and some experience before we started. I have to credit Eric Barton and Craig Forrest, who were graduate students with me at the time. Both are now assistant professors, Eric at Wake Forest and Craig at Arkansas. And they worked together to identify the receptor. And the strategy we used was absolutely dependent on having a non-sialic acid binding reovirus strain. Because if we used T3D, type 3 Deering, which binds to sialic acid, regardless of the strategy, we would have identified heavily sialated glycoproteins, and there are many, and who would have known which was truly a receptor? And the results would have been confusing because if they're heavily sialated, the virus would have bound to them, and it would have just made it difficult to sort out. So what we did is we used a virus that Eric generated called T3SA-, a type 3 reovirus that didn't bind to sialic acid. Mm-hmm. And we used a cDNA library generated from Entera2 cells. These are cells that were generated from a teratoma. And the idea is that you can treat the Entera2 cells with retinoic acid, and they'll differentiate into something that might look like a neuron. So we thought, okay, and they're human. And we thought that we wanted to identify a human molecule because we thought that there probably would be more reagents to work with if it was an unusual protein, if it was human rather than mouse. And we wanted to go after a neural receptor. That was the idea. We wanted the receptor on neurons. And so Eric and Craig set out to do this using an expression cloning approach, an approach that was pioneered by Arufo and Seed to identify interleukin receptors. And so we transfected into Cas7 cells the whole cDNA library. Now, Cas7 cells are not totally resistant to reovirus infection. They're a little bit susceptible. But Cas7s are transformed with T antigen so that any plasmid that has a T antigen origin of replication is going to be amplified in those Cas7 cells. So we transfect the library, and then we use fluorescinated T3SA-. So this is the ligand. It doesn't bind to sialic acid. It just binds to the definitive receptor. Now it's bright with a fleur. And then we use flow cytometry to select just the top 0.5% most fluorescent cells. We pull those out, open them up, collect the plasmids, retransform bacteria to amplify them, and then retransfect again, bind, and do this a series of 4 times. And then after the 4th sort, we're starting to see like a little shift, like the shoulder is starting to shift. So then Eric and Craig, and I think if they were here, they would say Eric provided advice and Craig picked about 1,000 bacterial transformants, as I recalled, and kept them in pools of 50, and then screened those pools of 50 and identified a couple of positives, and then took the pools of 50 and made pools of 5 and found a couple of positives. And sure enough, in 2 of these pools of 5, we found the cDNA encoding junctional adhesion molecule A, or JMA. Now, this was 99 2000 in that time frame, and at that point,
you're leaving us, Dickson. You'll be back. Okay, thank you.
That's my wife.
No, that's the important call to answer.
I'm sorry.
Yeah, no problem. So this is about 2000,
right.
And the first thing we do is go and do a little literature search to see what we can learn about JAM, a junctional adhesion molecule, and it had been cloned about a year before we identified it by a woman called Elisabetta Diana in Milan, and she of course cloned the mouse homolog of JAM, and so we do. This brilliant idea to pick the human cDNA library, and we didn't have any reagents. Well, it turned out that on the website of Chuck Parkos, who's a GI pathologist at Emory, he had just cloned the human homolog and wrote in his research description that he had identified this molecule, had monoclonal antibodies, and so I called Chuck and asked if he would send antibodies because we had made this finding. We wanted to validate this putative receptor, and he sent his panel of monoclonal antibodies, and sure enough, all of them blocked binding. So our strategy after that, once you do some type of a genetic screen to get a candidate, is all about the validation. And I think of the Koch's postulates of receptor identification.
Yes.
And I think that there are three. And so one is that if you inhibit the binding using an antibody, for example,
right.
that should inhibit the binding of your ligand, of your viral ligand, and that should
Right.
inhibit infection. The second is that if you genetically manipulate the expression of the receptor, either introduce it into a receptor-negative cell line or silence it with an SI or use a receptor-negative cell line, that manipulation ought to affect binding and infection.
Right.
And then the third, and I think the most stringent, is to show a direct interaction between the viral ligand and the receptor itself with a KD that makes sense, so a physiologically
Right.
significant KD. And we're lucky with reovirus because, of course, the ligand is this long filament which is amenable to expression in bacteria, so we can express the action domains and purify them. And GMA is an Ig superfamily protein, so we can express its ectodomain too and purify them both and show that they bind with a KD in the low nanomolar range.
Yeah.
So we thought, okay, so we've validated the receptor, but this still is sort of at least conceivably, a contrived in vitro cell culture kind of thing. So we were really anxious to do experiments with mice that lack JAM-A. Now, JAM-A is a fascinating protein. When it was first cloned, it was just JAM. Actually, when it was first cloned, it was called the F11R because there was an antibody to platelets called F11, and its receptor was the F11 receptor, of course. And it wasn't until Diana identified the molecule that it got its name JAM. And then soon after, there was discovered to be a family of JAMs, and so now there's JAM-A, JAM-B, and JAM-C. Of those three, only JAM-A is a reovirus receptor. And so it's expressed in tight junctions in epithelia and endothelia. It's also expressed on hematopoietic cells, and it's broadly. It's in virtually every tissue where those cell types are found. And so, stunningly enough, mice that lack JAM-A are viable. They give rise to fertile offspring, and so we can infect them. And so, imagine our excitement when we inoculated these animals orally with reovirus, and with an industrial strength dose, and the wild types all die with encephalitis, and the mutants, the ones that lack JAM-A, all survive.
Wow.
So we look at the loads, and the loads in the intestine of the knockouts are a little bit less, and there's almost no virus that we recover at any site of secondary replication. So then we wonder whether JAM-A is in some way promoting dissemination or whether it serves as a receptor at these secondary sites.
Yeah.
And it's the former, because when we inoculate virus intracranially directly into a site of secondary replication in the brain, then in the presence and absence of JAM-A, the virus infects just fine. So the virus does not require JAM-A to infect the brain. But it does require JAM-A to get into the bloodstream and to infect endothelial cells. And so that's its function. So here we started, these years ago, trying to find a neuroreceptor for reovirus, and we found a dissemination receptor for reovirus. And so I'm real excited because we have a new screen that we've done, which is a completely different screen. We did an siRNA screen, and this was work of Bernardo Mino, who I mentioned before. And Bernardo, with Jen Konopka and a new student, Danica Sutherland, identified a really interesting molecule in the nervous system that we're working to validate now. And so perhaps that's the definitive docking receptor that reovirus uses in the brain. But
JAM-A you got from a differentiated cell, which you thought might be neuron-like. Yeah,
correct.
It sounds, but it's an endothelial protein.
Correct.
So is it also present in the brain?
It is, but it's present on endothelium, so vessels, but it's not present on neurons.
Cells.
Yeah.
Okay, so that's probably why you cloned it out of the neural induced cell.
Yes.
Right.
Yes.
COS7?
Yeah.
What's that?
The COS7 cells?
Yes.
That was, by the way, quite a risk to use COS7s if they had a little bit of Rio binding activity. Because when we did polio, we used mouse cells, which have no binding at all.
I think what made it go was that these cells the COS7 cells express so much of the plasmid-driven insert,
perhaps,
that we could find it. But
perhaps.
Yes. Mm-hmm.
So Brian Seed once, when I was at MIT, he was at Harvard, and we got some vectors from him once to do this, and he had a sheet. Maybe you had the same sheet that came with the vectors, and he explains them all and everything. And at the end, it's just handwritten by Brian. If you have any problems or questions, just drop dead, okay? Did you get that one? No.
I did not.
I still have it. It's in my
not.
file. I still have it.
Yeah.
So you have, and then the other thing you did was to make infectious DNA copies of reoviruses, which have 10 segments.
10
so
segments.
12 of the rotas.
Right.
And people had tried this for years, and no one could get it to work.
Well,
And what was your secret? You
yeah.
used 10 plasmids, and you put them in cells, and you got it to work.
Yeah. Well, in fact, there's a large group of double-strand RNA viruses, not just the Reoviridae viruses, which are the 10, 11, 12-segmented ones, the big ones. There are these birnaviruses, bi-RNA viruses that have 2 segments. Yeah. And the way forward was paved really by the birnaviruses. They had developed these plasmid-based rescue systems for the birnas before we had done our work with mammalian reoviruses. The folks who did this, Vic Vicaria, Egbert Munt, sort of shined a path on a way that might be possible for this to be done. But you're right, lots of labs had tried to rescue reovirus from plasmid by making cDNA copies of the gene segments and using an appropriate polymerase to get them expressed, and it just didn't work. So, I have to give all the credit here to Takeshi Kobayashi. Takeshi was a postdoc with my group. He's now leading his own team in Osaka. And he came to our lab with the idea that he was going to generate a rescue system for reovirus. And this was, 2003, 2004, in that range. And I thought, well, I certainly admire your courage. Let's talk about some alternatives, because a development of rescue for reovirus is not going to be specific aim 1.
Yeah.
It's going to be specific aim 3, and let's work on some other tractable problems so that you have a couple of papers so that you might have a job prospect when you're finished with your postdoc. And Takeshi, in his wonderful way, heard that and worked full-time on the projects that we developed, and then he worked full-time on developing rescue for reovirus. So what we did is we cloned full-length replicas of each of the 10 segments so that the 5' end was right juxtaposed to a T7 RNA polymerase promoter. In the 3' end abutted a hepatitis delta virus ribozyme. This was exactly the strategy that several in the negative strand RNA virus field had used to generate rescue systems for paramyxoviruses, for Ebola, for lots of viruses that had used this system. Yoshi Kaioka sent us the original plasmids that we used for this. We pulled out all the Ebola sequences so that we're only working with reovirus, but that's where we got the original plasmids. So Takeshi made them, and he got to deliver T7 polymerase, a recombinant vaccinia called RDIS-T7 from a colleague in Japan. This is not MVA-T7. This is a different replication-incompetent T7 vaccinia, and it's not completely replication-incompetent. Of course, you can propagate it in chick cells, but it doesn't infect very well in mammalian cells. And so I must say, Takeshi tried a lot of strategies where we altered the number of G nucleotides at the 5' end to increase T7 transcription, and we ultimately used the exact native sequence. I think that the trial and error of transfect first, then infect with the vaccinia expressing T7, or reversing that, trying a couple of different cell lines, and eventually got it to work. These great days in science, the day when Eric and Craig said, we know what the receptor is, and they just knew it. It was an IG superfamily member. It couldn't be anything else but the receptor. And then Takeshi shows me a gel indicating that he's rescued a recombinant reovirus, and we engineered our plasmids so that they had marker sequences in them that we introduced, so we knew that the virus was recovered from plasmid and not a laboratory contaminant. And so that was just an awesome day. But I'm asked a lot, what did you do that made it work? And I would say the T7 polymerase expressing vaccinia that we used is less cytopathic than MVA or any other T7. The cells that we ultimately use, the mouse L cells, that was the first generation of our rescue protocol, are the most permissive cells that I know of for reovirus. So they really propagate the virus well. And so surely, and then careful cloning, ensuring that the sequences are pristine, and we corrected a lot of RT introduced errors to make the sequences exactly authentic to correspond to our laboratory strain. But more than anything else, what made this go was Takeshi Kobayashi's effort. This is an example of faith-based science, Vince. So he would not give up. He believed he could do this. There was no reason he could think of that it wouldn't work, and he was not going to rest until it got done. Yeah. And so now, I mean, it is amazing that the rescue system was reported in 2007, and we've been able to use it to introduce mutations in almost every single reovirus gene segment, to introduce vaccine epitopes, to alter the virus to make better oncolytics, and our colleagues are continuing to even do more innovative things with reoviruses based on this. So it was really a very enabling technology, and it was just the belief of one person that I can get this done. There probably is some biology to it, and I'll tell you this, it's just fascinating. If you take our plasmids and express RNA transcripts with T7 in vitro and introduce those RNAs into a cell, virus will not be propagated. Yeah. You cannot rescue virus from the RNAs. If you do the same thing with blue tongue virus, now blue tongue is a 10-segment Reoviridae family member. If you introduce the plasmids in cells that have T7, virus will not propagate. But if you generate the RNAs in vitro and introduce the RNAs into cells, they will propagate. This is work of Polyroys. And rotavirus is still a challenge. Rotavirus has 11 segments, and there's still not a fully recombinant rotavirus rescue system. Now, there are systems, John Patton's work, he's able to silence the expression of given rotavirus genes and then complement with a cDNA and trans and then get a virus to incorporate that.
You can't put 11 segments.
You can't put 11 plasmids in or 11 RNAs in and get virus to recover. And I don't think it's just a question of the numbers. I think there's something biologically different about each of these viruses that govern how a replication complex, forms and functions.
He needs to have, the field needs someone with a lot of faith to get that to work.
Exactly.
There's no reason why it can't, right?
Exactly.
It's got to work.
Yeah.
All right. I have one more question because we have to wrap up because we need to go to dinner. And that is, this is a great system. You can do so much. So why are you starting to work on chikungunya?
Yeah, thank you. Well,
it just sounds funny.
I love the name,
Yeah.
We all do.
There are a couple of reasons. One is that our lab has been going now for, this is my 23rd year. And I've been so fortunate over the last decade or so to recruit really terrific postdocs to my group. And they've come and established a beachhead of scholarship and made a name for themselves in their particular field, and they're moving out now to establish their own laboratories. And some of these themes are themes that I may have been working on for some time, or themes that they started de novo in our lab. But I really thought it important for them to have their own group and their own themes, and know that they weren't going to be doing experiments that were going to be done by a graduate student in my group. And so I must say that was the prime reason. And then I got to thinking about, well, if I was going to change and work on another virus or maybe add another virus, I can't imagine not working on reovirus. There's always going to be reovirus experiments in the lab, but I don't know, in 10 years, maybe 80% of our work will be on chikungunya. It's about the opposite now, about 80 on Rio and 20 on chikungunya. And I just started to read, and I wanted to work on an RNA virus. The DNA viruses are complicated, and they're big, and many of them have many genes, and it's a challenge. As a simple country virologist from Nashville, Tennessee, to think about working on one of these big viruses, and I wanted something that already had a rescue system, so that there was an infectious cDNA that could be manipulated, produced a disease, had a good animal model, and would offer me opportunities to collaborate with colleagues at Vanderbilt, but also colleagues that were elsewhere that I just hadn't had the chance to collaborate with, but I really enjoy their company.
It might get you to Italy.
Well, or Sri Lanka, or the Seychelles, or India. I mean, there's so many places where you'll find chick. And so that attracted me to chikungunya. And of course, it's of interest now as an emerging pathogen, and there are many who are interested in the virus. We don't have antivirals there's a vaccine, but it's not one that's really approved for use in humans. It's not as attenuated as it might be, and so there are a lot of opportunities, both for discovery-based science, but also for science that will have direct application to improvements in human health. So I have to credit Mark Heisey and Tim Morrison, who got us started, and we continue to collaborate with. My student, Allison Ashbrook, is going out to Tim's lab in Denver to do some animal experiments in their BSL-3 facility. Allison and Lori Silva, the postdoc working on chikungunya, have both visited Mark's lab to learn about the virus. And then we're doing a lot of work now with Jim Crow and Mike Diamond to try and understand the human neutralizing antibody response to chikungunya. Jim is able to isolate monoclonal antibodies from humans, and we've got some neutralizing antibodies that we're characterizing and learning how they work, and this has been really exciting. And
Good.
so it's been a fun project.
It sounds like a good thing. It sounds
Excellent.
like you considered it really carefully.
You can do work with mosquito biologists,
It
too.
can.
Well, we have some colleagues who work with mosquitoes, and they're very excited about opportunities for sort of cross fertilization of chikungunya and
It's a fellow
mosquitoes.
in Paris, Marco Vignuzzi.
Marco Vignuzzi.
He does mosquito with chick.
Great
right?
stuff. Yeah.
It's good. And you moved to plus strand RNA viruses, which has to be good.
Well, they have proteases, but the proteases are embedded, and it's amazing. They have their scissor activity that comes out and can clip on either side, so they can get to work. You got to love it.
Looks good. Sounds good. And I look forward to. Well, you haven't published in Chick yet, right?
Not yet.
Well, when you publish,
yet.
then we can get
Well,
you back, and you can tell us what you're doing
Let
here.
me just
Great.
say that I'm honored to have been one of your professors.
Thank you, sir.
We should say that at the onset, when you arrived, Terry, when you arrived in my office, Dickson was here, and you recognized him immediately.
Of course.
As a professor you had here at Columbia Medical School.
Of course. Well, every medical school is unique, and every medical school has these little gems that are maybe idiosyncrasies of that particular medical school. We had the best parasitology course in the world, and Dickson, of course, was the course director of that course. I mean, imagine we almost had a month of terrific lectures and laboratory, and for someone completely enraptured in infectious diseases who was a biology major and loved ecosystems, learning stories of worms that would need to go through a maturation stage in a snail, and then you would drink them and they turn into this 3-foot thing that came out your leg. I mean, what could be better,
Just what we were talking about.
That's right. That's exactly right.
Dickson and I were reading a new book of his that he's written. What's the title of it?
People, Parasites, and Plowshares.
Oh,
And we just
neat.
read the Chikungunya chapter.
Neat.
And you may not know this, but Dickson and I have done a podcast about parasites now for a couple of years, and he tells all these stories, and people love it. They say he's the ultimate raconteur. And
So there you go.
that's how you remember him as well, right?
Right.
Oh, dear.
Okay, this episode of TWiV will be, as usual, on iTunes and at TWiV.tv. If you like it, what you can do for us is to go over on iTunes and subscribe, and also to leave a rating. That helps to keep us visible so that more people will find us. And as always, we love to get your questions and comments. Please send them to TWiV at TWiV.tv. Terry Dermody is at Vanderbilt University Medical Center, where he has so many titles. One thing we didn't talk about, you're director of the Medical Scientist Training Program, an MD PhD program, which didn't exist when you were a medical student, right?
It was really small, and I remember I was either a first or second year medical student. And I remember visiting with the director of the program or someone in program leadership. One of my classmates, Leslie Lobel, was an MD PhD student, earned his PhD with Steve Goff. And I talked to Leslie a little bit about it and thought, that might be interesting. And I met with this man and expressed an interest in learning more. I'm interested in hearing more about the MD PhD program because I have questions about whether it might be right for me. And his response was, to paraphrase, son, if you have questions, we don't want you.
Wonderful.
We want people who are clearly committed to this career. And at that point, I couldn't have made a commitment. It took much longer for me to decide that science was really a career that I wanted to pursue.
Thank you for joining us. I appreciate it.
It's my pleasure.
I know I tried to get you here a long time ago, and you promised, and so I'm glad you did it. Tomorrow you're giving a seminar, of course, and talking, but I'm really happy that you sat down with us to do a TWiV. So thanks very much.
A lot of fun.
Well, my pleasure. Thanks for having me.
Dixon Despommier is at verticalfarm.com, Trichinella.org, and medicalecology.com. Thank you, Dixon.
Thank you, Vincent.
It was kind of serendipitous, right?
It was great.
But since you guys knew each other, I figured
it was great.
You should be here as well.
It was terrific.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.